So here we have a muscle fiber, which is also known as a muscle cell or a syncytium, and it has a membrane called the sarcolemma, which has nucleuses and infoldings, which are known as T-tubules, and then it will branch off into a myofibril, which has a net of sarcoplasmic reticulum, and then in here we have our sarcoplasm, which has mitochondria in it. In our sarcoplasmic reticulum, we have many protein pumps for calcium ions, and then the myofibrils will go off into myofilaments, which consist of thin and thick. The thin is the actin, and the thick is the myosin. Here we have our I band, which is the area of the actin, the thin filaments. And then we have our M line, where the thick filaments attach. Our Z line, where the thin filaments attach. And then our H band, the thick filaments. And then the sarcomere, which is the area between the filaments, the two filaments. This is the impulse, and then the impulse will move through the axon of the motor neuron to the presynaptic cleft, and then acetylcholine will diffuse across the neuromuscular junctions. The acetylcholine will attach to receptors of the postsynaptic cleft, and then that will cause for the sodium ion channels to open. The sodium ions will go inside, and that will cause depolarization in the postsynaptic cleft, and then an action potential is generated at the sarcolemma. An impulse will pass through the T-tubule, and then calcium ion channels will open. Calcium ion channels will go in, and then they will attach to troponin, and then that will cause a change in shape, and then the myosin heads will bind to actin to form cross bridges. We have creatine phosphate, which will provide ATP with a phosphate to make ATP. And during muscle contraction, the I band will shorten, and the H band will decrease, and then the two lines are going to move closer, and the sarcomere will contract.